Welcome. The subject of the day is pulse oximetry. I know that some of you just woke up and are maybe having your first cup of coffee. For some of you, the day is almost finished and you're going home from work soon. Some of you might be staying up late just to watch us at our webinar today. Thank you all for joining us. My name is Anja Rode and I'm Global Product Manager here on Oximetry. The measuring of SpO2 and pulse oximetry is maybe the subject that we get the most questions about from our customers. This is the Oximetry Pro device, and this is the pulse oximeter that we are talking about today. Clip it on my finger. Oximetry is titrating the oxygen flow based on the current SpO2 measured on the finger or on the earlobe of the patient. At the first webinar a month ago, Dr. Antonio Corrado from Florence in Italy, he's the former chief of the respiratory intensive care unit. He talked about the importance of titrating oxygen, and I will quote what he said. Oxygen should be treated like any other drug. Too much might be as harmful as not enough. This makes the measurement of SpO2 that we in our oxygen robots use to titrate the dose of oxygen very important to rely on. I must admit that even with many years of experience working as a nurse on pulmonary wards and intensive care units, using pulse oximetry as a life-important parameter to determine how to treat my patients, I expect to learn a lot from the presentations today. Once again, I'm very happy that you are here with us. We have participants from all over the world. A warm welcome to all of you. I will briefly take you through the agenda of the next hour that we are spending together. Our first presenter here with us today is Walter Holbein. Walter is a Ph.D. in physiology. He's also the Chronic Business Group Director from Nonin. Good morning to you, Walter. Walter will start today's webinar by telling us more about how pulse oximetry is working and the technology behind the measurement. Then the next presenter, she couldn't be here with us today. So last week, we had the pleasure of having Boss Søvang visiting us to record the presentation of her study that is going to be published. Anna is a nurse and she is also a master in health science. And Anna will do a presentation on her study measurement, precision, accuracy for 5 types of pulse oximeters used for patients with respiratory diseases. Then Walter will tell us more about the accuracy and biases in pulse oximetry. And finally, I will do a short presentation about closed loop oxygen titration by Oximetry, explaining how and why the quality of the pulse oximeter readings is so crucial for us. And during the webinar, the Q&A section will be open, and a team from Oximetry and Nonin will be ready to answer the questions that you might have on pulse oximetry and automatic oxygen titration. At the end of the webinar, Walter and I will go through some of the questions. For your information, this webinar is recorded and you will be able to watch or rewatch it afterwards and also share it with your colleagues. So now I will give the word to you, Walter.
Sounds great. Thank you very much. All right, then let's get started. So hello, everybody, and welcome to the webinar. We're excited to really have this opportunity to discuss the current state of pulse oximetry. As Anja reported earlier, my name is Walter Holbein, and I'm the Chronic Business Group Director for Nonin Medical. So we're looking at the telehealth, chronic disease management, and remote patient monitoring business. So the objective for today is really to develop a comprehensive understanding of pulse oximetry and really how these advantages of choosing Nonin pulse oximetry to provide the best possible care for your patients. For you to develop the understanding, we'll dive into the following topics. overview of pulse oximetry. We'll dive into a little bit of the physiology and science behind oximetry, as well as the pulse oximetry accuracy studies. So how are we able to show our accuracy? We'll show a few examples of demonstrated accuracy in clinical results, as well as showing the key differences of Nonin technology. So what you can expect to learn today is what is the true gold standard for determining accuracy in pulse oximetry, and how leading companies like Nonin compare to the standard in challenging conditions. You'll also understand why pulse oximetry has historically been monitored from the finger, which is a key aspect to the Nonin technology, and demonstrated accuracy in conditions of low perfusion and skin pigmentation. So let's get started. So you can see right away where Nonin gets its name from, because it's short for non-invasive. So pulse oximetry is sometimes called the fifth vital sign, and this is why, and this is because it's such a simple and painless way to determine the effective oxygen saturation in the body and how that's being transported around the body, and how often the heart is beating. Because this is such a simple and effective way to determine these key aspects of health, we get asked questions sometimes around how different solutions affect the accuracy. And there are some conditions that can have some effects, like low perfusion, different skin pigmentations, patient size and motion, as well as nail polish. Therefore, it's really important to select a pulse oximeter that has that demonstrated accuracy in all of these conditions. So let's get started with a bit of the physiology. So oxygen is vital to the functioning of cells. If we think back to our high school biology classes, this is because of cellular metabolism. Oxygen delivery to the cells is that important indicator to make sure that the person is healthy. This oxygen saturation is an indicator of transport of oxygen throughout the body, and indicates that sufficient oxygen is being supplied to the body. And this is especially for parts of lung function. Oxygen binds to the hemoglobin molecule in red blood cells and is transported throughout the body in blood. So the arterialized, or the oxygenated blood that's coming from the lungs, is from in the arterial side, and the deoxygenated blood that's coming from the tissue beds, that's coming through the venous side. As oxygen binds to red blood cells, this changes the way that light interacts with those blood cells, and the more oxygenated a blood cell is, the redder it becomes. So in very simple terms, pulse oximetry is actually reading how red your blood is. Perfusion is the circulation of blood through the tissues. The image on the right-hand side is an image of a hand. When you look at this is about normal perfusion for a human. Areas of greatest perfusion have a darker, redder color to it, and the lower perfusion has more of that bluer tone. Therefore, when we look at where we're going to get the most signal and the best information possible, we're looking for the areas of greatest perfusion, and that's the finger beds that we know and love today. So when we get into the science of pulse oximetry is really about the interaction of hemoglobin and red blood cells as it interacts with different colored lights. So the summary of this is the image to the right, but I'll explain this a little further. These two different light sources are composed of red light, which we can see, and infrared light, which we can't. These two different light sources pass through the blood, and pass through the body and go to the photodetector. The photodetector is where we are receiving the information from the light sources, and that's how we use an algorithm that's specifically developed for our pulse oximetry, and that's what calculates our SpO2 measurement. The results of this is a number that provides useful information to the patient's medical condition. Really importantly, these sources of light are essentially our source of truth. So impure light sources can shift the calibration and affect the accuracy of these measurements. And this is most notably at levels that are really important for the health of the patient, at saturations below 95 and darker skin pigmentations that are in some people. Therefore, when Nonin puts together probes and pulse oximeters, it's very important to have the highest quality LEDs. That makes the difference in your measurements. So Nonin oximetry, as I've mentioned, really has that quality that we're known for. This is due to our 30+ years of experience in light sensing technology. When we first developed the pure light sensor technology, provides only the purest red and infrared LEDs to produce high intensity pure light spectrums. And this has been shown to eliminate variations in readings for consistent measurements, especially at critical SpO2 levels. These high quality sensors also ensure the accuracy is not degraded due to skin pigmentation or low perfusion. So let's now get into how Nonin conducts accuracy studies so you can understand how Nonin delivers those measurements to you. So these are two examples of pulse oximeters that you may be familiar with. On the right is our external OEM module, and on our right is the fingertip pulse oximeter. These are the two types of our pulse oximeters, and both of these have gone through the same level of testing. So let's see about a pulse oximetry study. So the true gold standard of saturation is actually measured by a co-oximeter, or also a blood gas analyzer. This is a picture of a very standard co-oximeter, and this is where we're getting the truth from. Our pulse oximeters are calibrated against the invasive blood gas machine to determine accuracy to standards and regulation claims. A co-oximeter requires a blood draw, and in most cases, an arterial blood draw. So when we talk about an arterial blood draw for our studies, we place a indwelling arterial catheter. This is done by a trained anesthesiologist under ultrasound guidance. So this minimizes any pain or discomfort from our study participants. We take for the average adult, normal saturation ranges from 95 to 100%. However, for patients that are experiencing compromised respiratory function, their saturation can be lower, and it's critical that pulse oximeters have the ability to read accurately in these situations. So we connect, for our studies, we connect participants to a anesthesiology machine, and this is how we induce hypoxia, or how we reduce the level of oxygen in a person's blood. We slowly take away oxygen and replace that with nitrogen, which is already around us in the air, and this is how we accurately produce different levels of saturation. We repeat this simultaneous measurement of pulse oximeter and arterial saturations multiple times over the course of a saturation range from 70 to 100%. And then we repeat these in multiple different patients to make sure that we're getting the most accurate reading possible. So what does this look like in an actual study, how does the data look? So first and foremost, I'm going to orientate us to the graphs that we're looking at today. So on the left-hand side, on the x-axis, so the bottom axis is time, and on the y-axis, the vertical axis, is SpO2. This is also going to be for arterial oxygen as well. On the right-hand side is a scatter plot. On the x-axis, we have arterial oxygen, so the oxygen saturation of our invasive blood gas measurements. And on the vertical axis, we have SpO2, so the measurements that we are getting from our pulse oximeters. So as you can see from this graph, we are measuring SpO2 over time. At around 46 minutes, which is around here, we take an arterial blood gas. This is where we are comparing our SpO2 to the blood gas, and both seem to be in alignment. Both are in the upper 90s. So we continue to desaturate the participant, and this can be seen as the SpO2 is dropping over time. This is our indication that that we are successfully removing the oxygen and creating that that understanding of blood being, or the oxygen being removed. This continues over the course of the next several minutes, and we continue to take arterial blood gases as indicated here at this plateau and here. These studies continue all the way down to an oxygen saturation of approximately 70%. Once we've hit that plateau, or the study participant decides to end the study, we replace the oxygen mixture with 100% oxygen and resaturate the patient up to a normal level, and that ends the study. And this is the study that we repeat on multiple subjects for multiple times, and we get our overall accuracy. I hope this provides you with a deeper understanding of how pulse oximetry studies are done and how Nonin demonstrates its accuracy even in challenging conditions. So this is a artificial example, but let's look at a real example, and let's look at Nonin's averaging. So Nonin's pure set technology uses an intelligent pulse-by-pulse averaging algorithm, and that is able to adjust for the patient's condition. So pulse set technology averaging automatically adjusts to the fastest response time that it can, of 3 seconds or faster, and there's no need to set a mode or setting in your system. In the example shown on the right-hand side, you can see the difference between a completely unaveraged SpO2, which is the dotted yellow line, and our pure set technology, which is the averaged SpO2. In this case, we are only, this averaging time only equals 1 second, which allows us to have the ability to respond to desaturations faster. And in situations like automatic oxygen titration within the O2matic system, this response time is key. And this is why with the intelligent pure set technology, this is the fastest averaging with the best identification of pulses for really true patient assessment. So that concludes the first portion of the presentation. And when we come back, we'll be covering biases, accuracy, and a little bit more about Nonin technology. Thank you very much.
Thank you, Walter. As I said prior to your presentation, a lot of this is knowledge that I have never heard of or anyone talk about in my previous career working as a nurse. I remember using the pulse oximeter that was available at the department and trusting the value that I saw on the screen. Maybe you can recognize this from your everyday work in your clinic if you are a clinician watching us today. Now I will show you a video I have received from the clinic. It's from the COVID department at the Hvidovre Hospital. I think it will start now. So what you see is two different sensors measuring two very different values of SpO2 on the same patient. The patient is a COVID patient. The video was anonymized, but I can tell you that the patient was speaking and in the background. So the point of this video is, what did the healthcare professionals do? They called me and asked if there was something wrong with the automatic and the pulse oximeter on automatic. And I think that's why that's because many of us tend to trust the pulse oximeter that we use the most. They also did an arterial blood gas to find out the true SaO2 on the patient, the arterial oxygen saturation. And this leads me to the next presentation from Anna Boss Søvang. That's covering exactly this subject, the precision and accuracy of 5 different types of pulse oximeters.
Hi, I'm Anna. I have been a registered nurse since 2010, and I have recently finished my master degree in health science. During the next 10 minutes, I will tell you about my study where I have investigated the accuracy and precision for 5 types of pulse oximeters used for patients with respiratory diseases. This study hasn't been published yet, so I will not go into all the details. However, I will give you some of the main results. Yes. First, I will tell you a little bit about my motivation for conducting this study, the objectives, and how data were collected. Then I will present the pulse oximeters and probes that we have investigated, the main results, and in the end, I will try to give you some takeaways. Pulse oximetry are of great clinical importance. However, some studies suggest that the pulse oximeters are more inaccurate used for a non-healthy population. So I was wondering how I have always relied on the SpO2 value when I was titrating the oxygen supplemental for patients with respiratory diseases. What if the pulse oximeter is overestimating the SaO2 value? Well, then we can cause the patient to have hypoxemia. On the other hand, what if the pulse oximeter is underestimating the SaO2 value? Then we can cause the patient to have hyperoxia or even hypocapnia regarding patients with COPD. And all these conditions could be fatal. Therefore, my primary objective was to examine the accuracy and precision for commonly used pulse oximeters at the Department for Respiratory Medicine. And in addition, I wanted to investigate the association between the SaO2 level and the difference between SpO2 and SaO2, the bias. Secondly, I wanted to examine the agreement between 2 pulse oximeters by comparing 2 SpO2 values. And finally, I wanted to investigate the diagnostic properties for each pulse oximeter for detecting hypoxemia, defined as an SaO2 below 90%. Yes, so this procedure was relatively simple. When patients were to have an ABG as part of clinical routine treatment, we measured SpO2 from 2 different pulse oximeters. And when we had a stable reading, we obtained the arterial blood sample. And simultaneously, we recorded the 2 SpO2 values. And then we did this comparative analysis between SpO2 and SiO2, where we have a negative bias, which implies that the pulse oximeter is underestimating the SiO2 value, and a positive bias implies that the pulse oximeter is overestimating the SiO2 value. And this comparative analysis was also done between 2 pulse oximeters. Yes, for a better understanding, I will just show you the pulse oximeters and probes that we have investigated. First, we have Criticare, where we did 54 observations. Then we have Philips with 52 observations. And then we examined 3 Nonin probes used with automatic. We had 60 observations with the blue finger probe, 21 with the disposable probe, and 25 with the ear probe. Yes. And here are the results from the comparative analysis between SpO2 and SiO2. And as you can see, the Nonin disposable probe, the ear probe, and the finger probe all have positive mean biases, and therefore overestimated the SiO2 value. And if we look at the ear probe, it had the highest mean bias. So in average, it measured above 3 percentage points too much. Yeah. Then on the other hand, the Nonin with the blue probe underestimated, and the Criticare underestimated a little as well. However, this was statistically insignificant. If we instead look at the standard deviation, SD, we can see that Criticare had the highest standard deviation. And this implies that Criticare was the most imprecise. If we look at the targets here, illustrating precision and accuracy, you can see number 1 is precise and accurate. Number 2 is still precise, however inaccurate. Number 3 is accurate but imprecise, and number 4 is both inaccurate and imprecise. And we could say that Criticare corresponds to number 3, and the others are somewhere between 2 and 4. And this is just to illustrate how both imprecision and inaccuracy can cause an uncertain measure. Yes. Here are the results from the comparative analysis between paired SpO2 values. And you can see the 4 combinations that we have examined. And all these mean biases were statistically significant. And therefore, bias isn't only patient dependent due to the non-healthy population. Each pulse oximeter seems like it has a unique bias. Yes. Finally, we did this diagnostic test, and sensitivity refers to the pulse oximeter's ability to correctly detect hypoxemia, defined as SiO2 below 90%, and specificity refers to the pulse oximeter's ability to correctly reject hypoxemia when SiO2 were 90% or more. And again, the Nonin with the disposable probe, the ear probe, and the Philips had low sensitivities and high specificities. And I have to say that some of these results are based on few observations below 90%. But I do think that this analysis provides an important perspective. Okay, so the Nonin disposable probe, the Philips and the ear probe have many false negatives and few true positives. If we look at the Nonin blue probe and the Criticare, they have relatively high sensitivities and specificities, and therefore they have few false negatives and few false positives. And if we look at the diagram, you can see how you could actually move this limit for a more optimal cutoff value. However, I will not elaborate that part of the analysis. Yes. Here at the final slide, I will try to give you some takeaways. First of all, SpO2 is an estimate for SaO2. And you cannot expect the pulse oximeter to be perfectly accurate and precise. However, if we adjust for this a systematic over or underestimation, you could greatly improve the accuracy. And finally, we have to be aware that each pulse oximeter has its own unique bias, and therefore we cannot just randomly use different probes without considering them having different biases. Yes. That's it. Thank you.
Thank you, Anna. It was very interesting to hear about your study, and it really makes me think how patients can benefit from healthcare professionals being aware of this possible differences between two types of sensors. I also have an agreement with Anna that if you have questions to her, then you can just send them in the Q&A, and we will send them through email to Anna, and she will respond to them. I also know that we have participants and customers from all over the world. And another question I often get is related to skin pigmentation. I will give the word back to you now, Walter. And I know you're going to tell us more about the accuracy and biases when measuring the SpO2.
Perfect. So thank you again for this opportunity to talk about this. And I'm so thankful of Anna's study to look at her effects in actual patients. So it's a fantastic next step here. So for the first thing to really think about is, why are we talking about this? And it's clinically important that bias should be considered when monitoring patients when their saturations are below 80%. And this is really to ensure that you can trust that measurement, and that you, above all, that the patients, even in these most challenging conditions. So I'm going to give a few examples of this throughout the next portion. So all co-oximeters read slightly differently, so all different brands. So it's the only true way to have an apples to apples comparison of pulse oximetry technology is to compare devices against the same subjects with the same oximeter. The information that I'm going to be presenting today is based off a peer-reviewed published study with the three major oximetry manufacturers, including Nonin, with a variety of subject demographics, as you can see on the right. These were compared from actual blood gases and compared to the measurements with our pulse oximeter. So the bias is the mean difference between the oximeter reading and the SaO2 values from an arterial blood gas, as mentioned previously by Anna. So a positive bias means that the test oximeter overestimates saturation, and a negative bias means that the oximeter underestimates saturation. The Nonin PureSat oximeter with the clip sensor was compared to the other manufacturers, also with their clip sensors. All three products meet regulatory accuracy, so that is a very important distinction to make. All of these studies, all of these pulse oximeters had that accuracy measurement from the regulatory standpoint, but they all differed in their bias when it looked at the SaO2 values. Based off of the results, the Nonin PureSat clip sensor had the lowest bias compared to the gold standard, or the blood gas, throughout the oxygen saturation levels, as well as for all skin pigmentations. In the most challenging condition, at the lowest saturation level, which is the 70 to 80%, and in the darkest skin pigmentations, the bias of the Nonin PureSat oximeter with the clip sensor was at its minimum of -6%. And this was in contrast to the other manufacturers. In this study, Nonin's readings biased slightly below the blood gas reference, which may ensure greater patient safety, as higher readings may run the risk of failing to identify patients and providers of the status that could result in clinical concern. As I mentioned earlier, bias is that mean difference between oximeter readings and co-ox. Precision is the standard deviation of the difference between the co-oximeter measurements. So therefore, accuracy when it comes to pulse oximetry, measured using the term ARMS, is a standard method for reporting pulse oximetry accuracy, which combines both the bias and precision measurements into one simple term. As mentioned previously, all three of these products met their regulatory accuracies, however, had different biases. So an RMS error for SaO2 is used for the Food and Drug Administration, so FDA for the US, for device approval. Anything above 3% is not acceptable. Yet in this study, we have a few examples of some of these manufacturers having biases or ARMSs above 3. But focusing on the Nonin PureSat oximeter with the clip sensor, this was more accurate compared to the gold standard or blood gas throughout the oxygen saturation levels for all skin pigmentations. Nonin was actually the only clip fingertip oximeter that provided accuracy readings ±2 for what we're reading here, reading tested individuals at all saturation levels, regardless of skin pigmentation, which is below the FDA acceptable limit. This study concluded that the Nonin technology with the clip probe tested did not show any effect of patients with all skin pigmentations tested. So Nonin took this data just a little bit further and really took it to, what does this mean to a clinician? So Nonin evaluated the study data at saturation points commonly used for clinical decision making by medical professionals or clinicians. In this first slide, we're looking at a saturation level of 88%, which is commonly used as a clinical cutoff for interventions. So with the Nonin technology, more patients were correctly identified for oxygen delivery at this intervention point. For all patients tested, so in this condition, Nonin identified 91% of patients at the SaO2 intervention point of 88%. And in the dark skin patients tested, Nonin identified 97% of the patients at this SaO2 intervention point. This is really important for making sure that when patients need that critical oxygen the most, that we're accurately showing that there is a problem. So another key clinical cutoff and used in some clinics is 92%. 92% mimics very similarly with the data that we just showed for 88%, where Nonin now identifies 82% of all patients tested at this cutoff. And in the darker skin pigmentation, we identified 85% of these patients. This is really important with the Nonin fast pulse by pulse averaging. Now we're going to be able to not only read the patients at the specific cutoffs that are needed, but we're going to detect that earlier because we're averaging faster than the rest. As this data indicates, we are more likely to show that desaturation and identify those patients at those critical cutoff points. So you may be asking yourself, why are we discussing this? Why is this accuracy study so important? For one, the technology in the system helps in diagnosing and treatment of patients with potentially severe medical conditions. When the patient's lives are at risk, we want to make sure that we're identifying these patients and that using a pulse oximetry technology that has both demonstrated accuracy and is cleared by regulatory associations. Your system that's in your hospitals and your clinics are there intended to work on all patients. Given its recent highlighting in medical journal articles, skin pigmentation does affect pulse oximetry and can affect the clinical environment when it comes to pulse oximetry. So it's very important that you're using a technology that doesn't degrade and has proven accuracy in these patients with different skin pigmentations. Not all patients are healthy, and the patients most at risk, they're counting on the system to provide those treatment and care decisions. Oxygen saturation really is an important indicator of the patient's health. So we want to make sure that the devices that we're using are also accurate in the presence of motion, as well as low perfusion. And lastly, with a patient's status that can change so rapidly, and with the technologies now available with automatic titration like the O2matic system, we want to make sure that when something changes, that we are identifying those problems quickly and managing them accordingly. So this is really important, and this is why you have to select a pulse oximetry technology that has demonstrated accuracy time and again in challenging conditions and on all skin pigmentations. And that's why accuracy does matter. And the last is for key takeaways for this presentation. From the release of the world's first fingertip oximeter, Nonin has created this durable, accurate, and reliable measurement for all patients, regardless of skin pigmentation. There are 4 key differences that I want you to understand about why Nonin is different. And that's, we have quality sensors, low perfusion filters, that true pulse detection, and smart averaging. We discussed earlier, Nonin's accuracy starts with the quality sensors. All pulse oximeters use red and infrared light to measure SpO2. Only the PureSat sensor technology uses the purest red and infrared light to produce high intensity light spectrums, which eliminates variability. The PureSat technology also offers low perfusion filters that provide a true and reliable reading cleared by the FDA. This filter automatically removes noise from the weak or low perfusion signals to obtain a reading even in challenging patients. PureSat technology also features true pulse detection, which reduces false readings, locating the true pulse in low perfusion and motion. So with motion, we're able to remove these artifacts, reducing the time it takes to get a reliable measurement. And what this means to you and the patient is reliable measurements that are both consistent, trustworthy, and good quality. Nonin's PureSat technology is also equipped with the pulse by pulse smart averaging. And this is where we're talking about the fast averaging that is either 3 seconds or even faster. This saves time, as well as there are no modes to set an intelligence that automatically adjusts the averaging for challenging conditions. The combination between pure light and pure set technologies provides high responsive, precise system for accurate and reliable readings, even under the most challenging conditions. These are the key aspects that help create the Nonin difference. So I want to thank you, and I want to thank Oximetry for allowing me to speak here today. You now have a clearer understanding of true gold standard for accuracy measurements in pulse oximetry, and how Nonin compares to those standards in challenging conditions. You also now understand why pulse oximetry has historically been monitored from the finger, as well as showing some key aspects to Nonin technology with demonstrated accuracy in low perfusion, motion, and skin pigmentation. Thank you very much, and I'm going to hand it back over to Anya.
Once again, thank you very much, Walter. And I guess you are wondering why is all this important for us in Oximetry. So let me tell you more about how our device is, or our devices is using the pulse oximeter reading to titrate the oxygen flow. Some of you might have seen this before, and for some of you, it's the first time. But what you see on the left side of the slide is our Oximetry Pro device. This is an intelligent device that, through an advanced algorithm, is titrating the flow to the oxygen, the flow of oxygen to the patient every second to keep the SpO2 on target. And this target of SpO2 is one that the healthcare professional have chosen for this individual patient. The SpO2 is measured continuously on the finger or the earlobe, as we just heard about, of the patient, and by a Nonin pulse oximeter. And that's what you see on the right side of the slide. When the oxygen is manually titrated with a flow meter on the hospital today, the SpO2 is measured sporadically. There can be hours between the measurement of SpO2 and adjustments of the oxygen flow. There are studies showing that there's a risk of SpO2 dropping during sleep, and there's a risk of undetected drops in SpO2, not only during sleep, but also during a busy shift with lack of staff. There is a risk of over and underdosing the oxygen. I would say that there is a risk we are treating. What just happened there? Sorry. Can we go back? Yeah. Sorry. There's a risk that we are treating our patients with our hands in front of the eyes, only catching a glimpse of the patient's SpO2. If we compare manual and automatic titration at the hospital, then with manual titration, there is a possibility of the patient receiving an incorrect dose of oxygen and having an SpO2 outside of the desired range of 4 hours without knowing. Think again of the hands in front of your eyes. With the automatic and automatic titration of oxygen, the SpO2 is measured continuously, and oxygen is also adjusted every second if needed. Automatic will also start alarming if SpO2 is below the target. When the patient is at home and receiving long-term oxygen therapy, there can be months, even up to half a year, between the SpO2 is measured and the dose of oxygen is adjusted. This means that the patient receives the same amount of oxygen during rest, during activity, and during sleep. With our home device, Oximetry HOT, that you see on the screen, we introduce a personalized treatment based on several measurements and adjustments every day. If you attended the last webinar, then you heard clinicians share their experience with using Oximetry, both during the COVID pandemic and at pulmonary wards. But we also have a lot of data from the clinic. And what you see here is a graphic explaining the analysis of 1,000 hours of data from COVID-19 patients being treated with Oximetry at a COVID ward in 2020. We have learned during the last 1.5 years that COVID patients admitted to hospital in need of supplemental oxygen can be very sick and very difficult to treat since their clinic. If you again think of the last webinar and the expert Charlotte Sandau, she told us about the happy hypoxia as an example. The patient is very different from other respiratory patients. If you again look at the 1,000 hours of data, then the researchers found that every patient needed 2 significant flow changes every hour. And that means 48 manually adjustments every day for every patient. You also met the chief physician, Jan Dahlberg from Kolding at the last webinar, and he told us about the implementation of Oximetry at his department in the middle of a COVID pandemic. And he told us that he had calculated that every patient needed to be visited 50 times every day by the nurses. But after the implementation of Oximetry, it was reduced to only 10 times per day. The 1,000 hours of data also shows that 60% of the patients, they were stabilized, which means that they have SpO2 on target within the first minute of treatment. And that was also the experience from Kolding and Jan Dahlberg. I will quote him: "After minutes or up to half an hour with Oximetry, the patient is normally stabilized in the saturation interval you chose. It means a stable patient." At the end, I will show you how it looks when a patient is treated by Oximetry. And don't worry, I will explain this to you. This is a graph showing the SpO2, the oxygen saturation of a patient with COPD being treated for almost 20 hours. You can see on the left, the target interval for SpO2 is between 90 to 94. And the graph is showing all the fluctuations of SpO2 that was during the treatment. If you now look at the graph below, that's the oxygen flow that the patient received adjusted by Oximetry. And on the left, you can see that this patient was prescribed with 0 to 6 liters of oxygen. You can see how many adjustments that was made during the 20 hours. And I can tell you that this is even aggregated data. That means that you only see adjustments for every 5 minutes and not every second. As an Oximetry is adjusting the flow every second to keep the patient stable. And Oximetry is also titrating the oxygen flow to the lowest amount of oxygen. And as you can see, this patient was actually stable. He was kept on the desired SpO2 for nearly 90% of the time. That's time spent on target. From our study on COPD patients from 2018, we know that nursing staff can only keep the patient on the desired SpO2 for 47% of the time when the oxygen is manually titrated. I just told you that this was a stable patient, but I remember last year I met an interviewed patient being treated with Oximetry with the pulmonary fibrosis. When he were resting and asleep, he needed 3 liters of oxygen per minute to have a stable oxygen saturation. But as soon as he started moving in the bed or started talking, his need increased to 10 liters per minute. And this was an adjustment that was almost impossible to handle for for the nursing staff, it was almost impossible to titrate him between 3 and 10 liters manually. But it was done by automatic, and he was so happy that he asked me if he could get automatic back home for his long-term oxygen therapy. But the patient you see here, the COPD patient, he is resting in his bed. And I also think that you can imagine how it would look if he started walking or he was doing rehabilitation exercises. And rehabilitation using automatic is actually the topic of our next webinar that we are hosting in November. The date and time will be announced on LinkedIn and Twitter and through our distributors very soon. I hope that you will sign up. What I want you to take home from today is that there can be a bias between different types and brands of pulse oximeters. SpO2 is not always the same as SaO2. If you are in doubt of the SpO2, consider an ABD. And automatic oxygen titration can assure a more safe and efficient oxygen therapy than manual titration can. And just to repeat what Antonio Corrado from Italy told us, oxygen should be treated like any other drug. Too much might be as harmful as not enough. Thank you for your time and for listening. I hope you learned something new from today's presentation. And hopefully you have also asked a lot of good questions in the Q&A section. And in a moment, Walter and I will go through some of them. I can see that we are almost on time, but I hope that you have 10 minutes to spend with us with the questions and answers from the Q&A. Hi again, Walter.
Hello. Thank you for your great presentation.
Likewise, we're just waiting for the first questions to pop up. I haven't had the time to see what questions we have. Oh, it's for you. Hi, Walter. Great presentation. How often would you recommend switching fingers in regards of finger pain and numbness?
That's a great question, and thank you for asking. This is a common question that we get asked at Nonin. The nice thing about Nonin technology is that we'll give you accurate readings on each of the fingers. So this is a patient preference. If they're starting to feel pain, feel free to change the finger, and this can be done as often as you like. Nonin will read accurately with each of the fingers.
Thank you. Oh, I heard about a Bluetooth version of Nonin sensor. Is it possible to use automatic with a wireless sensor? I guess that is for me. At home, for our home device, we use a Bluetooth sensor since we don't want the patient to be hooked up on a finger clip sensor all the time. And the home device has a more advanced algorithm that only needs measurement from time to time. But at the hospital, we use the wired sensor to be sure that the sensor is not running out of battery or is getting exchanged with the patient in the bed beside. So for patient risk safety, or for sake of the patient safety, we use the wired sensor. It's more stable and the staff can be very busy. So yeah. Let's see if we get another question. Oh, can SpO2 measurement be affected by altitude?
I think that's another great question. So SpO2 is affected by altitude, not from the standpoint of its accuracy being affected, but the oxygen concentration at altitude is reduced. So you would expect a SpO2 to read lower than lower at altitude than at sea level, and this can range all the way down into the lower 90s as well, depending on how far you are in altitude. Our oximetry has been used for Everest Base Camp research studies, which is an exciting opportunity to measure real world SpO2.
Wow. Yeah, we have any more questions? How does automatic do when there is no signal from the pulse oximeter? That's also a very good question, and thank you for that. If the pulse oximeter is off the finger or the signal is lost, then automatic will continue with the same amount of oxygen as the last value. And after at a maximum of 5 minutes, there will be an alarm on the device. So it is possible for the patient to change the placement of the sensor or sanitize their hands. If we get it. Why do certain sensors read differently than others? Oh,
I think this is a great question for me. Thank you, Anya. This is a nice follow-up to Anya's study. All oximeters go through the same clinical evaluation as I mentioned previously. So each one of those sensors and oximeter combinations have gone through a similar breathe-down study, and each one of those cases, their overall accuracy meets the regulatory requirements. With that said, there are certain aspects that may affect the readings one way or another, but in all cases, as Anna pointed out, they may read a little differently. However, they're going to still give you the information you need as long as you take that into account.
Good. I think we can take one more question and then I think we should round up. Oh, what is the minimum SpO2 level that can be measured and treated by automatic and Nonin? I read this as two different questions because I know that you can measure a lower SpO2 than we are treating. Automatic has a treatment interval for patients between SpO2 80 to 100. And if the SpO2 is below 80, then it will start alarming. But you can choose your treatment interval between 80 and 100. So how low can you measure a stable SpO2 with your sensors, Walter?
So our accuracy studies are measure SpO2 and SaO2 down to a oxygen saturation of 70. We do have some data even below that, but our accuracy is measured between 70 and 100.
Thank you. And we will, of course, also show the value of 70 or 66 or whatever it is on the screen of automatic. So it's not like it's going blank. We are using your sensors and we'll follow what the value that they give us. So thank you all for being here with us today, and thank you, Walter, for the great presentations. I must say that I have learned a lot, and to you, Anna, thank you for the presentation of your study. I'm looking very much forward to reading the article when it's published. As I told you in the beginning, the webinar was recorded and will be available on YouTube together with the first webinar and in this series about automatic oxygen titration. And you can rewatch both or share them with a colleague that you think could benefit from this also. I hope to see you all again in November when we have this webinar on rehabilitation. And I'm very excited because we are going to share some really good results with you. So thank you all once again and have a nice day.
Thank you.